We're Clara, Evianne, and Rebecca, and our video is going to be about in vitro fertilization. First, I will be explaining the basics of IVF, such as the history, rules, and procedure. Evianne will then talk about the role of Kisspeptin in IVF, and after this, Clara will talk more about the recent developments within the fertility field. So, what is IVF? IVF, or in vitro fertilization, is a type of assisted reproductive technology, or ART, in which the oocyte gets fertilized outside of the body, or in vitro, so in glass. It is used to treat infertility and is used in case of surrogacy, in which a woman bears a child for other individuals. IVF has been around since 1978, when a few doctors fertilized a woman using natural cycle IVF, which means that there were no drugs used to overstimulate the ovaries, and which means that the success completely depended on the normal ovulation of the woman. In the end, Louise Brown was born without complications, and the doctor who made it all happen received the Nobel Prize of Medicine in 2010. So when are you eligible for IVF? IVF is used to treat fertility for patients with blocked or damaged fallopian tubes, ovulation disorders, male infertility factors such as sperm counts or abnormal sperm, prior tubal sterilization or removal, genetic disorders, endometriosis, a condition in which the uterine tissue starts growing in other places like the ovaries or fallopian tubes, and for patients with unexplained infertility. But are you eligible for IVF if you just have one of the previously mentioned physical problems? No. In order to be allowed into the IVF program, women have to meet a certain amount of requirements. These differ for every country, but the most universal rules are that you need to be quite old. In the Netherlands, for example, most clinics will refuse to help people under the age of 40, and that you have to have tried to conceive for at least 1 year during the fertile periods of the menstrual cycle. So how does IVF work? This has already been explained by our guest lecturer Meerte Smits, but here is a short recap of how it all goes. First, hormonal drugs are prescribed to induce ovarian hyperstimulation, which means that multiple oocytes are being prepared or matured for ovulation. This is usually done with injections. After this, egg retrieval can start. During this step, the mature oocytes are taken out of the ovaries by a needle that extracts the fluid in the follicles that contains the oocytes. Around 8 to 15 eggs are successfully retrieved on average. Then, the oocytes are fertilized. This is done with either frozen sperm or donor sperm. After this, all that is left is the embryo transfer. This is done with a so-called catheter that places the embryo after a few days of development in the cervix.
As described earlier, oocyte maturation has to be triggered to cause ovulation for the collection of the ovums. Oocyte maturation is caused by the LH surge that is caused by the positive feedback from estrogen on the Kisspeptin neurons. In the IVF treatment, 2 hormones are currently used, namely HCG or gonadotropin-releasing hormone agonists. HCG is used since it's structurally similar to LH and works on the ovarian LH-HCG receptor in the granulosa cells. LH or HCG is the most common trigger for oocyte maturation. However, due to its prolonged activity, it can cause ovarian hyperstimulation syndrome. Ovarian hyperstimulation syndrome shows symptoms of enlarged ovaries, ascites, hydrothorax, renal failure, and can be life-threatening. Moreover, women with polycystic ovaries have a higher chance of developing severe ovarian hyperstimulation syndrome. Therefore, another treatment has been developed, namely gonadotropin-releasing hormone agonists. These agonists have a shorter duration of action and thereby cause less stimulation, and therefore can limit the risk of ovarian hyperstimulation syndrome. These agonists bind to the gonadotropin-releasing hormone receptor in the pituitary and causes an increased secretion of LH and FSH, resembling the LH surge in a normal menstrual cycle. Previous research has shown that gene expression of ovarian hyperstimulation-related risk factor genes are upregulated after HCG treatment, compared to the gonadotropin-releasing hormone agonist treatment. Risk genes include vascular endothelial growth factor, Cadherin-5, and platelet endothelial-derived factor. A couple of years ago, another treatment was proposed, namely Kisspeptin. The underlying mechanism could be explained as follows. We can see that if the levels of estradiol reach a certain threshold, Kisspeptin neurons are activated. These neurons release Kisspeptin 54, which regulates the hypothalamus-pituitary gonadal axis. Kisspeptin binds to the Kis1 receptor in the hypothalamus, which stimulates the release of gonadotropin-releasing hormone and the LH secretion in the pituitary. Therefore, it was hypothesized that Kisspeptin could also induce oocyte maturation. In 2015, Ibarra et al. Showed that Kisspeptin 54 triggers oocyte maturation in women at high risk of development of ovarian hyperstimulation syndrome, and that the treatment had successful oocyte maturation and pregnancies without any significant development of ovarian hyperstimulation syndrome in the mothers. Since there is a difference in gene expression between HCG and gonadotropin-releasing hormone agonists, it's interesting to determine the effect of Kisspeptin 54 on the gene expression compared to the previous treatment. Furthermore, Kisspeptin receptors are also expressed on the granulosa cells themselves, and therefore might play a direct role in stimulating the ovaries. Therefore, in this paper, Owens et al. Wants to assess the in vivo effect of Kisspeptin 54 on the gene expression and compare it to HCG and gonadotropin-releasing hormone agonists gene expression patterns. And secondly, determine if the Kisspeptin effects are primarily effective on the HPG axis or also has a direct action on the human granulosa cells. The study showed that the FSH receptors were 14-fold more expressed than previous treatments. Similarly, the LH receptors were 2-fold higher expressed. The mean rise in the levels of FSH was 2.4 milli-international units per milliliter, and the LH was 55.4. Nonetheless, most women in the Kisspeptin group had polycystic ovaries and therefore are likely to have higher levels of FSH and higher expression of FSH receptors in general. Therefore, when these are correlated, it can be seen that these are not confounded, and therefore the upregulation can be explained by the Kisspeptin treatment. Steroidogenic hormones that were upregulated after Kisspeptin treatment were STAR, 3-beta-hydroxysteroid dehydrogenase 2, ESRR1 and ESR2, and inhibin A. There was no difference in gene expression of markers of ovarian hyperstimulation syndrome between all groups. Granulosa cells treated with Kisspeptin 54 did not show different gene expression compared to the control. To conclude, the study shows that Kisspeptin 54 treatment alters the granulosa cell gene expression considerably, especially compared to the traditional triggers. This is not visible in vitro experiments. This overall demonstrates the indirect effect of Kisspeptin via the HPG axis. Also, ovarian hyperstimulation syndrome markers are not upregulated upon Kisspeptin treatments. More interestingly, the serum levels induced by the Kisspeptin treatment resembles the physiological menstrual cycle more than the more traditional triggers, and the gene expression pattern potentially favors an ovarian environment for the progesterone synthesis and implantation. Limitations of this research are that the granulosa cells used for in vitro experiments are already exposed to Kisspeptin and thereby could have influenced the results. So further experiments could involve ovarian biopsy to collect follicles and expose these to Kisspeptin treatment. Also, most women in these studies had PCO. Therefore, further research could demonstrate the same effects of Kisspeptin treatment in healthy females with different fertility problems.
So up till now, we've heard IVF is really promising for infertile couples that want to have a child. But guess what else you can do with IVF? You can do something called 3-person IVF. But what exactly is this? As a parent, you usually want the best outcome for your child, and I'm not only talking about future careers and education, but health. You want your child to be healthy, and while this can be done when the child is growing, for example, the child eats healthy food, it exercises, it stays away from smoking and other drugs that could be bad for the health, there is always a chance your child is born with a disease, sometimes unforeseen or untreatable before the birth. But what if I told you that there is a way to decrease the chance of your child being born with a specific disease, namely mitochondrial diseases? And what has this to do with IVF? So to quickly revise, mitochondrial diseases are a group of diseases ranging from mild to severely life-threatening caused by errors in the mitochondria. We know the mitochondria are super important organelles as they generate energy, or ATP, to be used by many processes in the body. So you can imagine that if something is wrong with the mitochondria, problems can arise in multiple systems of the body, such as the gastrointestinal system, musculoskeletal, endocrine, heart, and even central nervous system. But if there's something wrong with the mitochondria, can't the cell itself fix it? The problem resides in the fact that the mitochondria have their own DNA, coding its own proteins and processes. We inherit this DNA from our mom's own mitochondria and not dad. The nucleus in our cells does code for some proteins needed by the mitochondria, but it cannot alter the inherited mitochondrial DNA. Therefore, if a mother knows she has some defects in her mitochondria, all her children will inherit this defect and increase the chance of developing mitochondrial diseases. So how can you prevent your own child from this terrible fate? This is where 3-person IVF kicks in. Basically, in this process, your child ends up with healthy mitochondrial DNA that is not the mother's. But how does this work? There are 3 components necessary: the father's nuclear DNA, the mother's nuclear DNA, and a healthy donor's egg. So, step 1. You take the healthy donor's egg and remove its nuclear DNA. You make sure you keep the mitochondrial DNA. Step 2. You remove the mother's and father's nuclear DNA and insert it into the donor's egg. Step 3. You allow the fertilized egg to develop a little before being transferred into the mother's uterus, just like in vitro fertilization. Now you have for sure a healthy embryo developing into a fetus inside the mom. So 3-people IVF is only legal in the UK, and currently the Australian government is weighing the scientific and ethical issues and how to best regulate this technology before it can be legalized. But if it were up to you, would you want to legalize this technology? You are reducing the chance your child is born with a defect, so this could be the best way forward for a healthy family. So to take home, we'd like to say again that in vitro fertilization is an assisted reproductive technology in which the oocytes gets fertilized outside of the body and is used to treat infertility. Kisspeptin can be used as a trigger for oocyte maturation and has no upregulation of OHSS risk genes, resembling the physiological menstrual cycle, and has a direct effect on the HPG axis. And finally, in vitro fertilization can also be used in 3-person IVF to prevent a child from being born with mitochondrial diseases. So, if you are infertile, don't abandon hope, because IVF can give you a healthy child after all.